Hello biology students, and welcome to video 6 of the organic chemistry unit. So in this video, we're going to continue our discussion of proteins, and more specifically, we're going to look at how proteins fold and also some various functions of protein molecules. Okay, so remember from last video, we said that a protein we defined as a polypeptide chain. So remember, a polypeptide, that was the polymer. It was the string of amino acids linked together, okay? So a protein's a polypeptide chain that's been folded into some shape, okay? And it turns out that this shape, whatever shape the protein folds into, that shape determines its function. So let me give you an example. So there's a protein in your cells called myosin, and it's shaped something like this, okay? So basically what it has is the protein has a little section on top that can attach to stuff. So maybe it's carrying, an object. And then it has two little structures that kind of look like legs. And these two structures literally walk along a different protein. So, these legs will swing back and forth. And it walks along this other protein called actin and carries its cargo through the cell. So in this case, if the protein didn't have the shape, if it didn't have this little spot to hold on to something and the two little legs for it to walk, it wouldn't be able to perform its function, which is to transport things around the cell, okay? So it turns out proteins can lose their shape. If a protein loses its shape, it's called denaturation. It has denatured. And this happens usually for one of two reasons. Usually if there's heat around or chemicals that interrupt the bonding, the interactions between different parts of the polypeptide chain. Okay, so we'll look at those in a little more detail on the next slide. So here's a picture of denaturation. So here you have a normal folded protein, and maybe this protein, its shape's really important because maybe whatever it bonds to bonds right in this little hole. And if that hole wasn't there, it couldn't bond to whatever it's supposed to bond to, and it couldn't do its function. So if we had too much heat or too much of the right kind of chemical, right now, maybe this protein is folded the way it is because the amino acid right here is attracted to the amino acid right here. So, there's an attraction there and it's causing it to fold. And maybe there's an attraction between this amino acid here and this one here, causing it to fold like that. And what happens is the heat or the chemicals we add interrupt these bonds, causing the polypeptide chain to go back to its linear original form. And this is useless to us because this can't perform the job because it no longer has that particular nook where it would bond to something. So obviously, the way proteins fold and the shapes they obtain is very important. So we're going to look at that folding process now. This can be a little confusing for students, but I think you guys can get it. Okay? So first, there are 4 different levels of protein structure. The first level is called primary structure, and that's just the order of amino acids in a polypeptide. Okay, so for example, in this one over here, I have, a threonine amino acid followed by a glycine, a glucine, a serine. And if I switched up this order, I would say that I've changed the primary structure of that protein. Secondary structure you can think of as the first fold, so the very first fold a protein makes. Tertiary structure is when those folds fold on one another to make even more folds. And quaternary structure is where you get multiple polypeptides to come together to form a bigger molecule. So let's look at each of these in a little bit more detail. The first primary structure, this is just the order of amino acids in a protein, the amino acid sequence. And this sequence is dictated by your DNA, okay? Sometimes this primary structure is called beads on a necklace because it's literally, if we draw amino acids like little circles, it's literally just the order of beads, just the order of amino acids in a protein. And it turns out if you change just one amino acid in a protein, you can totally change how the protein will fold down the line. So an example, if there are several hundred amino acids in a protein called hemoglobin that carries oxygen in your blood. And if you change just one of those amino acids to a different one, the protein folds differently. That hemoglobin can no longer carry oxygen, and you get what's called a disease called sickle cell anemia. So this order, this primary structure is very, very important. Okay, so secondary structure is when parts of the protein start to do their primary folding. They're the very first folds, okay? So these folds are usually due to hydrogen bonds that are formed between the amino group and the carboxyl group on different amino acids. And there are 2 types of secondary structure. They're what we call alpha helices and then beta pleated sheets. So let's look at some examples here. So you can see, here's my primary structure, my order of amino acids in my polypeptide. But you can see we've started to do some very basic folding. You can see there's kind of a little fold right here in the blue. This is called the beta pleated sheet. Okay, and you can see what's happened is, if you look, I have one amino acid here, and you can see I formed a little hydrogen bond between the oxygen on the carboxyl group and the hydrogen on this amino group. Okay, so you can see I have kind of like one side is hydrogen bonding with another strand, and it's forming that first basic fold. So that's a beta pleated sheet. It's the very first types of folds that come when a protein's folding. The other type of secondary structure, the other basic type of folding we can get is an alpha helices or alpha helix. A helix is like a spiral. So you can see, imagine that this section right here formed a spiral shape because we were getting, again, hydrogen bonds between carboxyl groups and amino groups on different amino acids. Okay, so the next step is tertiary structure, and it's basically when the whole protein, all of the alpha helices and beta pleated sheets start to fold on one another. So it gives the overall 3D shape. So you can see here, I have some alpha helices, right? So some helixes. I have some beta pleated sheets. But really, the whole thing is folding on itself, okay? All the alpha helices and beta pleated sheets are folding on one another. So this is tertiary structure. And this is created by hydrogen bonds, just like secondary structure. But it also can be caused by ionic bonds, and then hydrophobic and hydrophilic R group interactions. So you can think, if my amino acid had a hydrophobic R group, that would want to be in the middle of the protein, somewhere. You can imagine this protein is surrounded by water because it's in your cell. So all the hydrophobic groups want to clump in the middle, and all the hydrophilic R groups want to clump on the outside because they like water, and this kind of helps the protein to give its overall shape. And there are tons of different shapes you can get in tertiary structure. Globular means kind of like a glob. Fibrous means long and thin, like a fiber. Okay, so that's tertiary structure. And then quaternary structure is when you get more than one polypeptide to come together. So maybe I have one tertiary structure that's all folded, it's over here, and a second tertiary structure over here. Now, if these two came together and started interacting, that would be quaternary structure. Okay, so not all proteins do this. Only some proteins have more than one polypeptide. And some examples are hemoglobin, that's the one I mentioned earlier that carries oxygen in your blood. Collagen as well. Okay, so I really like this picture because it just reiterates a point that I made earlier in the video, that protein shape is really important for its function. So for example, here I have an unfolded protein, a protein that's been denatured, and its amino acids are, all in a nice line. But when this folds properly, due to secondary and tertiary structure, now you can see these amino acids are all together, and maybe, just the way they've lined up, they're able to bond with something. So now this specific setup of this little binding site allows this other molecule to come in and bind. So it's showing how this specific folding can allow it to bind or interact with a secondary molecule. Okay. So let's, real quick before we get into protein function, let's talk a little bit about how if you eat a protein, how it's broken down. So proteins are broken down in the digestive tract, so in your stomach and small and large intestine. So, and as they're broken down by hydrolysis into amino acids. So those amino acids are going to enter your blood, and the first thing that's going to happen is cells are going to take them out of the blood, and they're going to use them to build new polypeptides and new proteins. Okay. So then any that are extra, so that's the first step, step number 1. But sometimes you might have some extra amino acids. So what happens to these extra ones is the amino group is first removed. This is called being deaminated. You can kind of see that amino, that root is in there, so we're de-amino grouping it. We're deaminating it. And we remove it because it turns out a lot of extra amino group is actually pretty bad for the body because it can form what's called ammonia, which is toxic to the body, okay? So that amino group is removed, and that amino group is considered to be nitrogenous waste. Nitrogenous mean nitrogen containing, right? The amino group has nitrogen in it. And that's and the body gets rid of that via urea, which is the main component of urine, okay? So we get rid of those amino groups that we removed. And then the rest is formed into a glucose-like substance, and it's used in cell respiration. Remember, cell respiration is how we use glucose to make energy. Okay, so just to reiterate, first we use amino acids to build new proteins, but any extra ones have their amino group removed, and that amino group is flushed out of the body as nitrogenous waste, and the rest, the glucose-like substance, is used in cell respiration. So finally, the last thing I want to do in this video is really quickly talk about the different jobs or functions that proteins can have. There are a ton. Know that proteins do almost all the work in your body. So first, there are transport proteins. These are proteins that transport things around the body. So the example I've already mentioned a few times now is hemoglobin. This is the protein that carries oxygen and carbon dioxide in the red blood cells in your blood. And I mentioned earlier, if for some reason, the hemoglobin in your body does not have the right primary structure, it doesn't have the right amino acid sequence, you can be anemic, you can have an iron deficiency, or you can even have sickle cell anemia. Okay, so in sickle cell anemia, I already alluded to this a little bit earlier, but just one amino acid in the whole protein sequence is changed to something else, and therefore, because it's changed, the protein does not form properly, and it can't carry oxygen. So a normal red blood cell with normal hemoglobin is nice and round, and can carry oxygen really easily, where if I change just one amino acid in that protein, the blood cell now is sickle-shaped, okay? And it has a hard time flowing through blood vessels because it's not round, and it has a hard time carrying oxygen. So a second function of proteins, kind of the one you probably think of all the time when you think of protein, is contractile. It allows your muscles to contract and flex, okay? And the two main proteins that do this are actin and myosin. We have defensive proteins, proteins that attack foreign things in your body, so things like antibodies. And so if you get a bacterial infection or a viral infection, you have certain proteins that go and try to kill those foreign invaders. We also have structural proteins, so things like keratin, collagen, elastin. So these are the main proteins that make up your hair, fingernails, and skin. Okay, so they provide a structural component to your body. We have signaling proteins. So we've already seen a couple of these. These are things like hormones. So remember from the carbohydrate section of this unit, remember insulin and glucagon? Those are the two proteins that signaled the pancreas signaled the liver to tell it to either store or release extra glucose as it was needed in the body. Also things like growth hormone. This is the hormone that tells your body that you need to grow larger. We also have storage proteins. Okay, so storage proteins are just storing excess energy. So you can think of like the protein in an egg white. In theory, that protein was storing energy for that developing egg if it ever got fertilized. And then finally, enzymes. We are going to look at enzymes for quite a bit. The entirety of the next video is going to be on enzymes. And these are proteins that speed up chemical reactions. Okay? So that's it for video 6, and I will see you in video 7.